Hello, my name is Frankie Zheng Xiaohao. I'm from Group 2, Subgroup 3.
Hi, my name is Ahmad Tayyip Azamuddin Khalid and my metric number is 97219.
Hi, I'm Clarence and I'm from Group 2, Subgroup 3.
Hello, I am Dayang Wahida binti Abang Zainuddin, Group 2, Subgroup 3 and matrix number 97354.
Firstly, 4 ml of distilled water were placed in 6 test tubes using micropipettes. Secondly, 2 ml of Bradford reagent were placed into each tube. Thirdly, 50 microliter, 100 microliter and 200 microliter of BSA protein standard were then added in duplicate. Fourthly, mixture in each test tube were mixed using a vortex. Finally, the samples were read from the least concentrated and the absorbance for each was recorded. A sample without protein was used as the blank to zero the machine. In part 2, the same procedure was repeated by replacing BSA protein standard to unknown samples A, B, and C.
In experiment 2, we have learned the Beer-Lambert's law. We learned how light absorption is directly proportional to the concentration of a substance and how this principle allows us to determine the concentration of biological molecule like protein using absorbance measurement. We also developed hands-on instrument handling skill by working with a spectrophotometer. This includes calibrating it using a blank solution and measuring the absorbance of samples at a wavelength of 595 nanometer. Another key area was the preparation of standard solution. We have gained a practical lab experience in pipetting, preparing standard protein solution and using them to create a calibration curve, which is essential for quantitative analysis. Finally, we also improve our data analysis skill. We learn to calculate average absorbance, determine standard deviation, and use the linear equation derived from the calibration curve to accurately estimate the concentration of unknown protein samples.
I will be sharing the main challenges in conducting protein analysis using spectrophotometry, and I will explain why it can be difficult. Spectrophotometry is a technique used to measure how much light a solution absorbs. In protein analysis, it helps us determine the concentration of protein in a sample, usually by comparing it to a standard curve. First major challenge is accurate sample preparation, especially when preparing the standard protein solution. We must create a series of solutions with very specific concentration of protein, usually through serial dilution. Even a small pipetting errors like drawing slightly too much or too little can lead to incorrect absorbance readings. Since the accuracy of our unknown sample relies on this standard curve, one small mistake can affect all the result. Maintaining consistency is another challenge. If cuvettes are not clean or if different pipettes are used for each sample without proper rinsing, cross-contamination or light scattering might be happen. This can lead to absorbance values that do not reflect the actual protein concentration that can reduce the reliability of the data.
Here are my 3 main suggestions to improve this experiment for future session. First, we can provide more protein concentration like 5 nanogram per ml or 25 nanogram per ml. This helps create more details of standard curve, lead to a more accurate result. Second, automated pipetting system might be helpful to reduce human error when sample preparation. Consistency and reliability of the experiment may be improved. Lastly, software tools like Excel for data analysis really good for calculating average and standard deviation. The graph plotting can be improved and streamline the data processing.